Thanks everyone for coming to listen to this talk. I'm sure this is not your area of expertise, so I'll try and keep it simple for you to understand what we're doing with mosquitoes. So our team is actually working on developing new techniques for surveillance of major mosquito-borne disease vectors, and we focus mainly on Zika, dengue, and malaria, and we work with partners in Africa, Brazil, and Australia. Okay, so mosquitoes are actually considered the deadliest animals in the whole universe. They kill over 700,000 people every year, and that's followed by humans, just under 500,000 people. Crocodiles come in 10th, killing about 1,000 people a year, and sharks and wolves, they just kill 10 people. I don't know if that's true in Australia, because sharks kill a lot of people in Australia, but that's the statistics from the Gates notes. And malaria is actually the deadliest of the mosquito-borne diseases. It's transmitted by female Anopheles mosquitoes, and it kills over 400,000 people every year, and 90% of these deaths actually occur in sub-Saharan Africa, 90% of the deaths occurring among children under the age of 5 years. And over 300 million cases are reported each year, and the disease affects over half of the world's population. Dengue is the second deadliest disease. It's transmitted by mosquitoes of the genus Aedes, and Aedes aegypti is the primary vector, and Aedes albopictus is the secondary vector. It affects between 50 and 100 million people every year, and dengue cases have been rising up for the last 5 decades. And it's endemic in 100 countries. Australia is one of them. Outbreaks have been reported in the northern part of Queensland, and half of the world's population is at risk. There's no vaccine for this disease, and there's no drug therapies. And what about Zika? Zika is actually an emerging arbovirus that was discovered in Uganda. Uganda is a very tiny, I don't have a pointer. It's a very tiny country in East Africa. I come from Kenya, so Uganda is just a neighboring country to Kenya. And it was discovered by scientists that was supported by the Rockefeller Foundation, and they're trying to understand the salivatic cycle of yellow fever, and then they discovered the virus in sentinel rhesus monkey. And then they inoculated this virus into mice, and then they realized that the virus was actually different from yellow fever. And that's how they knew that that was a different virus, and they named it Zika virus from Zika forest. The virus was then recovered from Aedes africanus, which is the jungle mosquito. And the first human case was reported in Uganda and Tanzania 3 years later. The disease is transmitted by the same mosquitoes that transmit dengue and yellow fever and chikungunya. Zika then spread to the equatorial Asia between 1969 and 1983. And the first major human outbreak was reported in the Yap Island in 2007. And then in 2013, the virus spread to the Pacific islands of New Cook Islands, New Caledonia, and Easter Islands, Solomon Islands, causing a major outbreak in those islands. And in 2015, Zika spread to South America. 2016, Brazil recorded about 7,000 cases in just over 2 months. They still didn't know it was Zika virus until they discovered that babies were born with small heads, and then they did more research and realized that Zika had actually found its way in South America. And in 2016, Zika made the United States at home as well. So there have been about 5,000 cases of travelers returning to the US from affected areas, and over 200 cases of Zika in the US that were locally transmitted in Florida and Texas. And there have been over 35,000 cases of local transmission in the US territories, including Puerto Rico, US Virgin Islands, and American Samoa. Zika virus was then declared World Health Emergency of International Concern in February of 2016, and until then there have been close to 500,000 suspected cases, and half of those have been confirmed to be Zika. And over 3,000 cases of microcephaly that have been linked to Zika have actually been reported since 2015. And like we mentioned before, there are no vaccines for these diseases. The vaccine that are being developed is mainly for malaria, but it's not even effective. It lasts for 6 months and there are relapses. There's no drug therapies for arboviruses, and resistance in drug therapies for malaria are widely spreading, and therefore vector control is the only way of managing all these diseases. And what are some of the things that we're using to control these mosquitoes? Because Aedes aegypti is a primary vector for dengue, Zika, chikungunya, yellow fever, we mainly try to target the vector so that we can manage all these diseases simultaneously. And Aedes aegypti likes to breed around people's homes, so if you have containers with water lying around your homes, these mosquitoes will come and lay eggs there and make their home there. So we try and tell people to eliminate breeding sites around their homes by just tipping pot plants or water storage containers. And we also talk to people to tell them how they can reduce the source of mosquito breeding sites. And this is one of the biological control methods we use for killing larvae of Aedes aegypti. So this is an insecticide that is put in resting sites of mosquitoes. So when mosquitoes rest in it, they pick up the insecticide and then they will fly to the breeding sites and they kill their own babies. We call it suicide bombers. And then we have bed nets and indoor residual spraying. This is mainly used for malaria control in Africa. Bed nets kill mosquitoes that are coming to bite people while they're sleeping, while the indoor residual spraying is mainly used to kill mosquitoes that rest on the wall. So when mosquitoes feed, they obviously want some downtime. They don't want any disturbance. So they go rest on walls, and if the walls are actually sprayed, they pick up that insecticide and they get killed. And we also kill mosquitoes by enticing them into something like that box there. So that box there, we put something that smells like a human being, so synthetic odors. The mosquitoes will think that there's a human being in there, and then they fly inside. Obviously, we put something toxic that will kill the mosquitoes, a bit like the Red Wedding in the Game of Thrones. So once they're in there, they get zapped or they get killed by an insecticide. And on the right-hand side here is toxic sugar baits. So mosquitoes also like to feed on sugars. So one way we can kill them is poison the sugar and make the sugar available to them, like readily available to them. So if they see this one compared to the flowers where they will have to extract the sugar, they will prefer to go to this. Only problem with this is it will target other species like bees, and yeah, we don't want that. And there are newer vector control interventions like Wolbachia. Wolbachia is a bacteria that affects 60% of insects. Unfortunately, this bacteria doesn't occur naturally in mosquitoes, especially Aedes aegypti, that transmits multiple diseases. So what scientists did is they extracted this bacteria from fruit flies, and then they micro-injected it into mosquitoes. And then they released the infected mosquitoes into the wild, so that when they meet with the wild, the bacteria is transferred to the mosquitoes in the wild. This is how it works. So if you release females that are infected, they met with the wild population that is not infected, the resulting population will all carry the bacteria. The second option is to release males that are infected. They meet with the wild population that is not infected, and the resulting eggs will not hatch. And then the third option is to release both males and females that are infected, and then you end up with a population that is all carrying Wolbachia. So it works in 3 different ways. It can suppress the population of mosquitoes, and this is where you release males, met with the wild population, no viable offsprings. And the second one is replacing the population. You release both females and males that are infected, or females that are infected. Once they met with the wild population, they will take over the population and all of them will be infected. The resulting population blocks the virus. So it's been shown that Wolbachia can block dengue, can block Zika, it can block yellow fever. And the third option is you release the life-shortening strain of Wolbachia, and what this does is once it gets to the population, it mates with the wild population, and it reduces the survival of the mosquitoes in the wild to a point where these mosquitoes cannot support the incubation period of the pathogen. So the mosquitoes are actually killed before they can transmit the disease. And last year, Brazil actually released Transgenic Aegypti produced by Oxitec. So what these mosquitoes do is they have a self-limiting gene, and when males are released in the field, they met with the wild population, and the offsprings die before they become adults. And that's what was actually used in Brazil. And guess what? Brazil declares end of its health emergency of international concern, and they claim that this happened because of the GMO mosquitoes that was released. And because the GMO mosquitoes were targeting Aedes aegypti, the number of chikungunya, dengue also dropped. And so how do we know if these control interventions are effective? We have to do surveillance. So surveillance tells us a few things. Tells us where the transmission hotspots are, so that instead of just doing blind spraying, we know that this area is infected, so we will just target our resources in that area, so we don't waste our resources. If we do surveillance, then we can also tell if our interventions are not working. And if we do surveillance, we can figure out what sort of species are in the population, then we can use the right intervention. We can also tell if mosquitoes are infected and tell people to avoid that area. And something that I've been working on for a while is trying to tell the age of mosquitoes. So we do this because mosquitoes can only transmit an infection when they have reached a certain age. So we try to eliminate older mosquitoes so that they don't have that time required to support the incubation period of the pathogen they're carrying. We also do surveillance because we want to detect insecticide resistance before it spreads in the whole population. So like I just mentioned, mosquitoes actually need time to develop so they can support the incubation period of a pathogen they're carrying. So when a mosquito emerges, it takes about 2 days before it goes out looking for a blood meal. And once it finds a blood meal that is infected, it has to support that pathogen for about 10 days. And that means that mosquitoes become infectious when they're about 10, 12 days. So what vector control program managers want to do is they want to eliminate this population here, which is the infectious population. So all the vector control interventions are designed so that the age structure is actually shifted to the younger population that are not transmitting infections. And people use a variety of techniques to do surveillance. The one at the top corner there is ovary dissection, and this is when we put the mosquito under microscope and dissect its ovaries. We do that because we want to know whether the mosquito has laid eggs before. If it has laid eggs, it means that mosquito fed on someone, developed the eggs, and laid the eggs. So it could be potentially infectious. If the mosquito hasn't laid eggs, then that mosquito probably hasn't fed on someone. So that mosquito might be potentially non-infectious. The other technique we use is analysis of cuticular hydrocarbons, which also estimates the survival of mosquitoes. And we use ELISA to detect infections in mosquitoes. And gene transcriptional profiling technique is used for a variety of things, including age, species, and infection status. What's the problem with these techniques? Ovary dissection is cost-effective, but it only tells you whether mosquito has laid eggs or not. So it doesn't tell you really much. Cuticular hydrocarbon technique for age grading is very expensive, so most control interventions, especially in Africa, will not afford such a technique. Transcriptional profiling is also relatively expensive, and protein ELISA, very slow and expensive. All these techniques will just tell you or group mosquitoes into 2 or 3 groups, which doesn't tell us much really. So the near infrared spectroscopy technique is what I've been spending time to work on for the last 10 years. And what we want to do is try and see if we can use this technique as a cost-effective, rapid, and high-throughput technique for surveillance of mosquito-borne disease vectors. And so what's special about this technique? This technique is special to us because it's non-destructive and very simple to use. All we do is put a sample under a probe or a light, shine a light on the sample, get a spectrum, and analyze that spectrum for a variety of things. It does not require reagents to operate, which means it's cost-effective compared to PCR, where you need a number of samples and extraction protocol and all that. This technique doesn't need that. It's requires little sample processing and takes a very short time to get a spectrum from a mosquito. I would say 3 seconds, but because you have to transfer the mosquito to a reference panel and then direct the beam onto the mosquito, let's just say it takes about 15 seconds, which means you can scan up to 1,000 mosquitoes in a day if 2 people are working. Now, you can't do this with PCR. It's just not possible. And if you know what you're working with, if you know what species you're working with, if they're infected and what age you're working with, you can actually develop models to predict all these things simultaneously. So if you have models for age, infection, species, all you need to do is scan the mosquito once, and it will tell you if that mosquito is infected, how old that mosquito is, and what species the mosquito is. And so using this technique in a malaria context, we showed that it was at least 35 times faster than existing techniques like dissection or PCR, and it was over 50 times cheaper than PCR and employing someone to do dissections. So what have we achieved so far? So I will tell you a story of the application of this technique since 2007 until last year, and I'll briefly mention what we're doing in Brazil So the technique was actually introduced on mosquitoes 10 years ago by scientists working in CDC and USDA in America, together with scientists in Tanzania. So all they wanted to know is if this technique can actually tell the age of mosquitoes, differentiating these groups into those that are potentially infectious and those that are less harmful. And they also wanted to know if a population has two cryptic species of mosquitoes both transmitting malaria, can this technique actually differentiate these two groups? So the first result, this is the first ever publication on this technique for mosquitoes, and they showed that the technique can actually differentiate mosquitoes in different groups. But the problem is, because mosquitoes are very tiny and the physiology of mosquitoes doesn't change that much, we see these overlaps. Like day one mosquito can actually look like a 5-day-old mosquito. Now, there's nothing wrong with the technique. It's just that mosquitoes are too small, and telling the age of a mosquito is actually asking too much. So we decided to split the population into mosquitoes that are actually just potentially infectious and those that are less likely to be infectious. So we decided to split the population into mosquitoes that are less than 7 days, those that are over 7 days. So if you catch a mosquito that is over 7 days, it's likely that it's fed, and if you fed on an infected person, then that mosquito is potentially infected. So we showed that we can group the population into two groups with 84% accuracy. And these two mosquitoes are the major African malaria vectors. This work was done in Tanzania in a semi-field system. And so it was just a build-up on the first study, which was done in a lab reared environment. And then the problem came in. We don't always scan fresh mosquitoes, because the first two studies were done on fresh mosquitoes. We had to come up with a way of, if we are collecting mosquitoes in the field, we have to bring them in the lab and scan them later. So we had to test a preservation technique so we can apply this tool in the field. So the first thing I tested was the RNA later. And I scanned mosquitoes when fresh and preserved them and scanned them later. And I've shown that there was no difference between fresh and preserved mosquitoes. And because this technique was fairly new on mosquitoes, the questions just kept coming. And the people come to us like, we cannot afford RNA later, it's too expensive for us. So we decided to test other preservation techniques as well. And we recommended silica gel, where you just dry mosquitoes and then scan, or put them in the fridge, or an ethanol 95% wasn't so good. And fresh mosquitoes were slightly different from mosquitoes that had been put in that preservation for 50 days. So we don't recommend, or if you have to use ethanol, then scan them maybe after 2 weeks or 3 weeks. Or canoe solution was also good. And then because we have to use the tool in the field, mosquitoes in the lab don't look the same as mosquitoes in the field. So we had to test and see if this technique can actually be applied in the field. So what I did, I went out to the field, collected mosquitoes that are resistant to pyrethroids, because that's what we use in Africa for control of mosquitoes. So if mosquitoes are actually exposed to pyrethroids and are resistant to those pyrethroids, or are susceptible to the pyrethroids, does this affect the accuracy of the technique? And again, we showed that the accuracy doesn't change whether a mosquito is resistant or susceptible to pyrethroids. And then, so how do we apply it? This is just hypothetical case study. So the near-infrared can be applied in this situation. So assuming there's an area endemic with Zika or malaria, and you want to spray that area so that you reduce the survival of mosquitoes in that area, you will want to know if the age of mosquitoes in that area is higher before you do the spraying, and then check the age structure again after the intervention. So assuming mosquitoes are 8 days old before you do the intervention, and then you spray, and then that population is reduced to 6 days, will the infra-red tell you the same thing? Because that's what we really want to know. Because of the accuracy is not actually 100%. Is it going to mislead you, or is it going to tell you the truth? So the infra-red will actually tell you that population was 8 days. And it's true, the population was 8 days. And then you spray. Will the infra-red technique tell you that the mean age has actually reduced to 6 days? Now this is where the problem comes in. Infra-red has errors. So it will tell you, yes, the population has reduced, but there's an error of 1 day. That's okay. That's okay, given that the physiology of mosquitoes doesn't change that much. An error of 1 day is actually not that bad. So we actually, based on these results, we recommend that this tool can be used out in the field to evaluate control interventions for mosquito-borne diseases. So like I mentioned before, we wanted to know if we can simultaneously predict the age and species of African malaria vectors. So scan once and tell whether that mosquito is young or old and what species it belongs to. So this is just a table showing different studies we did alongside the age grading results, and we showed that infra-red was more efficient than PCR, simply because there was so many failed attempts for PCR to amplify the samples. So it was easier to use the near-infrared because you will always get a result. And if it's 80% or 90%, you at least know that 90% of these species are actually in this population. But if PCR is actually failing 50% of the time, you won't be able to know what sort of population you're dealing with. And for the first time, we introduced this technique on Aedes aegypti, which is a mosquito that transmits not malaria, but dengue, and it's found in canes. So we used mosquitoes that were collected in canes, and we wanted to know if we can apply it to predict the age of Aedes aegypti. And we have shown this was actually one of the best results we've ever got with age grading. And we showed that, yeah, we can tell the age of mosquitoes. And this is particularly important for the Wolbachia project, because if you're releasing Wolbachia infected mosquitoes, you want them to survive long enough so that they can invade the population. But if they're not surviving long enough, then the invasion is not going to happen. So then that intervention might actually fail. So we do age grading just to see, is this invading the population? Is it surviving long enough to invade the population? We also showed that we can tell the age of male mosquitoes. This is particularly important for interventions that release male infected mosquitoes, like not male infected, but male mosquitoes that are sterile, so that when they met with the wild population, they sterilize the whole population. Because you want these mosquitoes to survive long enough to mate with the wild population to sterilize the whole population. So knowing the age will actually tell you if these mosquitoes are surviving long enough. So we have shown, this is the only technique that has shown that male mosquitoes can actually be age graded. And for the first time, we also showed that we can detect Wolbachia in Aedes aegypti. And it was so good, although the males were less accurately predicted. This is just because Wolbachia is concentrated in the ovaries, and we think that because near-infrared is picking up the concentration of organic compounds in mosquitoes, and male mosquitoes don't have ovaries, that's why maybe it's finding it hard to to detect in males. Nevertheless, we showed that 83% of those mosquitoes were correctly predicted, and with females we got prediction accuracies of up to 96%. Now Rio is has these Wolbachia infected mosquitoes, and they've released them in the wild, and so our USAID project is also focusing on developing models that can detect Wolbachia in the field of Rio. And so the bacteria Wolbachia has two strains. So the life-shortening strain and WML, which is not the life-shortening. So we also wanted to see if we can differentiate mosquitoes infected with different strains. And here we show that we could differentiate a WML infected mosquito and a WML pulp infected mosquito with 97% accuracy for females and 85% accuracy for males. And I think that's pretty much what we have actually done and has been published. All our manuscripts are in the public domain, so you can have access if you want to know more about what we've done so far. And right now we are focusing on using this technique to detect Zika in mosquitoes in Brazil, and we're working with Fiocruz on this project. And like I mentioned, we are also using it to detect Wolbachia in the fields of Rio. And our future project, because we're using a lab-based instrument, we want to narrow down to a smaller handheld instrument. So we're trying to see if we can test handheld instruments for mosquito-borne diseases in multiple countries, and use this to connect to a smartphone so that they can be used on sites. Like just take the little handheld infra-red out to the field, scan a mosquito, and see if that mosquito is infected. And I'm pretty sure if we have such gadgets, everyone will want to have it. Like even you guys will want to have it. Like you want to protect yourself from infected mosquitoes out there. And we're also using it to see if we can detect nematodes in feces of sheep and goats, and also in grass. And this is being done by our PhD student Elise Koch, is sitting behind there. And Jill is also wanting to apply this technique to profile nutrients, and she wants to focus on humans and especially malnourished children in Africa. And that's our team, Global Integrated Disease Surveillance Team. We have our partners up there from the US, Floyd and Bob, and then Tom Churcher from the UK, and our Brazil team is just over there from Fiocruz. We also work with people in Kenya, Dr. Mata and Access Afia for Jill's project. And in Australia, I'm sure you recognize some of these faces here from QAAFI. And just to acknowledge our funding from USAID, Grand Challenges Canada in Canada, and CDC cooperative grants, and On Prime. And we got our instruments from USDA, provided to us by Floyd and Bob from CDC. And we work with Fiocruz, Army Malaria Institute here in Brisbane, Ifakara Health Institute in Tanzania, USDA in the US. And some of these projects were hosted at Kiwaima Berghofer here in Brisbane. And mentorship support is from Floyd and Professor Bob Wards, both from the US. And thank you very much.